Welcome students. Today we are going to study the microbial diversity with respect to the Sundarban ecosystem. Every microorganism has its own way of thriving and attaining nutrition for its survival, hence creating a diversity. In an ecosystem, the microbes play a very important role in degrading and thus recycling the materials of an ecosystem. An ecosystem consists of the living parts, that is the biotic community, and the non-living or the physical and chemical parameters that govern the biotic community, known as the abiotic community. Over here, we are taking an example of an estuarine ecosystem, where due to the high tide, the water reaches up to this portion, and during low tide, the water level comes down to this portion. Hence, in this ecosystem, the water inundates and recedes, creating a dynamic ecosystem. The Sunderbans is one of the largest deltas of the world. Over here, we have the Bay of Bengal in the south, the river Hooghly to the west, and Bangladesh in the east. The fresh water from the river comes and mixes with the saline water of the sea, thus creating a unique environment and thus diversity in the Sundarban ecosystem. The biotic component consists of the living things. In the Sundarban ecosystem, we have the tiniest of the producers, that is the phytoplanktons, to the largest of the predators, that is the Royal Bengal tiger. Microbial diversity of the soil of the Sundarban ecosystem mainly consists of bacteria, actinomycetes, and fungi. All of them are responsible for degradation and recycling of the material there. They are photosynthesizing microscopic plants that drift on the water, and habitat is the euphotic zone, that is the zone over here where the sunlight can reach up to, because photosynthesis requires sunlight. The depth of the euphotic zone is generally up to 200 meters where sunlight can penetrate. Hence, they occupy this euphotic zone of the ecosystem. They are the agents of primary production, that is, they convert atmospheric carbon dioxide and with the help of water and sunlight produce organic matter that serve as food for the rest of the food chain.
Sir, how do phytoplanktons act as the base for the aquatic food web?
As you can see here, the phytoplanktons are the producers.
Right.
They synthesize the organic matter from CO2 and water. The fish larvae feeds on the phytoplanktons, which in turn are eaten by small fishes, and the small fishes attract the larger fishes. In this way, a cycle goes on, and hence the phytoplankton forms the base of the food chain. Phytoplanktons can be classified into diatoms, cyanobacteria, and dinoflagellates. Generally, we have these three categories under which we can study phytoplanktons. Experiment to isolate and enumerate microorganisms from soil and water. Agar plate method and viable count method is the most widely used and common method for enumerating microorganisms present in soil sample. Over here, we get the number of bacteria or the colony forming units present in the plate, hence the number of organisms in the sample. We need suburban agar media or nutrient agar media or glycerol asparagine agar media. It's based on according to the type of microorganisms would be present and what nutrients they would require to grow. So we can use these three media which suffice our requirements for most of the known bacteria. Now we have the sterile Petri plates and sterile water and 9 ml tubes. 1 gram of the soil sample is dissolved in 9 ml of sterile water in a conical flask. It's agitated and mixed well and allowed to settle. Then 1 ml from there is taken and poured into 9 ml of distilled water in sterile tubes, giving us a dilution of 10 to the power minus 2. In this way, 10 to the power minus 6 dilutions are prepared. After the dilutions are prepared, we perform pour plating. In pour plating, we take 1 ml of each of the dilutions and 20 ml of the nutrient media and slowly pour it into the Petri plate. We allow the plates to solidify and then incubate them at 37 degrees centigrade for 24 to 48 hours, after which we take the readings or the observations. Over here you can see quite a number of bacteria have grown in this agar plate. After that, we need to isolate or identify the bacteria. For that, we use gram staining to identify the bacteria based on their morphology. This is a preliminary step for identification. For gram staining, we need a clean grease-free slide. We make a bacterial smear. Initially, we stain it with crystal violet. Then we wash it with water. We put iodine, then safranin dye, and again wash it with alcohol. Then we see it under a compound microscope, either in low power or high power, giving a magnification of 450x. There were two different colonies of bacteria present. One is the gram-negative Pseudomonas species and a genus of gram-negative motile rod-shaped Flavobacterium that were isolated from the soil of the Sundarbans. Both of these are salt-tolerant or halophilic bacteria. The plating should be done aseptically. Next, the plate should be inverted. Next, when we pour it, the nutrient medium should not be very hot, otherwise it would kill all the bacteria present in the sample. Phytoplanktons are diverse, and in order to study them, one should follow certain morphological traits that help us and makes it easy for us to understand these wonderful creatures. The best way to study phytoplankton is through a phase contrast microscope because of the contrast they provide that help us to see the morphology. Over here you can see that the phytoplanktons are diverse and each are different and have a specific morphology of their own. They can be broadly classified under kingdom Monera, that is cyanobacteria, blue-green algae, and kingdom Protista, the diatoms, coccolithophores, and the dinoflagellates.
Sir, I would like to know what importance do the phytoplanktons have in our lives?
Well, the phytoplanktons, apart from the food chain being primary producers, also help to absorb the atmospheric carbon dioxide because they are the photosynthesizers. Apart from the trees and plants, they absorb carbon dioxide and they sequester it. And after the phytoplanktons die, they settle down. Thus, the carbon dioxide are trapped. So, the pollution and the CO2 level can be reduced by these phytoplanktons. And also, 3/4 is water, and these phytoplanktons can be in the sea. So, we have a huge scope that the phytoplanktons can absorb CO2 if they bloom and are numerous in number. A clean grease-free slide, a dropper, a cover slip, a phase contrast microscope. Stain if required. Generally, stains are not required because of the high contrast. For specimen mounting, diatoms are put in a drop of distilled water into the slide. They are covered with a cover slip and glycerol is put. Then they are observed under the phase contrast microscope. If stain is required, Canada balsam stain or Bengal dye is there. The phytoplanktons can be identified based on the morphology or the structure. Over here, you see the flat discoid shape of many centric diatoms, example the Cytonella and the Cossinodiscus. The needle-shaped species of Rhizosolenia and Nidaria. The long, sometimes coiled filaments of some species of Belloceria. As you can see the bristles over here of Stephanodiscus and the horns of Chaetoceras. This is an individual Chaetoceras which are joined together. The Asteronella species, they form a colony by the mucilage linkage, and the Talaria species also formed colonies. They always stay in colonies unless or until they are broken away due to some conditions. Next, we have these extensive mucilaginous envelopes of Cytonella species. The cells may be sometimes joined by mucilage and forms a network. Colonies of cells may be embedded in a common tube and forms a tubular structure such as Nischia navicula. And the horns of Chaetoceras, they also are joined together by these special outgrowths and form a colony or a chain. The water sample, when observed from phytoplanktons in the Indian Sundarbans, revealed these phytoplanktons: the long horns of Chaetoceras, the grouped colonies of Asteronella, and the tubular form of Nischia. Make sure the slide is clean and grease-free and free of contamination, and use less background light in the microscope to reveal the special characteristic morphological features. Otherwise, they will not be intricately visible. The non-living or the abiotic components of an ecosystem consists of the physical and chemical parameters such as temperature, pH, salinity, sunlight, precipitation, and many more. All of these are very important and interconnected with the ecosystem and sustains the biotic community, which largely depends on them. We would be studying about the pH, salinity, electrical conductivity, and the total dissolved solids. These make up the vital components that are responsible for the proper sustenance of the biotic community. pH is the negative logarithm to the base 10 of hydrogen ion. It tells us how basic or acidic a solution is. In an ecosystem, its relevance is, suppose the pH changes rapidly from 7 to 9. What would happen? The phytoplanktons would die, and hence there would be a lapse in the food chain. The interesting part of the Indian Sundarban is that the pH varies from 7.2 to 7.9, which is quite a difference. This is because of the monsoon, pre-monsoon, and the post-monsoon. Due to the heavy rainfall, when more water comes in and the salinity decreases, the pH also changes. So the biotic community over there can adapt to this wide range of pH. Hence, it's unique in this ecosystem. Salinity is the measure of all the ions present in water. It can easily be measured by using a refractometer. As the name suggests, it refracts light due to the presence of ions, and hence we can get a clear and reading of the salinity by simply using a refractometer. The electrical conductivity can be measured in a very simple way using the conductivity meter. The concentration of salt ions when reaches a very high concentration can no longer be measured by this meter because it does not give accurate results. As you can see here, the electrical conductivity initially here but gives a saturation out here when the level of salt concentration is high. Can you say why?
So I think if you take for the estuarine environment in the Sunderbans, because they have a salty environment in the water, after more addition of salt, I mean saturation point will be reached, and hence like we already have an excess of molecules, and hence it no longer will increase the conductivity.
You are partly correct, but the actual point is when there are too much of ions, they form chelates or they form groups. Hence, they cannot conduct electricity in the way when they are individual. So, we don't get an accurate reading. The electrical conductivity can be converted to TDS, that is the total dissolved solids, by a simple empirical formula with a factor of 0.64 when multiplied to electrical conductivity, which is in microsiemens per centimeter, gives the total dissolved solids, which is in ppm. This is only an estimate and does not tell us anything about the nature of water. The total dissolved solids is defined as the amounts of organic and inorganic ions present in a water sample. So how does it help us to determine how the water is in an ecosystem, or what is the use of measuring TDS? As you can see here, acceptable borderline for a water is 200 ppm. Above that, we can say that the water is hazardous. Over here, we take an example of a fish. We see at ppm up to 300, the fish is smiling and is happy. But over here at 500 to 700 ppm, the fish is unhappy and is suffering. Hence, the simple experiment for total dissolved solid tells us about the quality of water. Is it habitable or inhabitable by the biotic community? pH is the negative logarithm to the base 10 of hydrogen ion. Most natural waters have pH between 5 to 8.5, which can sustain microorganisms and the biotic community. Water has a pH of 7, which is neutral. So over here we have a scale showing 7 as neutral pH, below 7 it is acidic, and above 7 it is basic. We need a beaker, portable meter, a glass rod, and a weighing machine if we take a soil sample for estimation of pH. Initially, we have to calibrate the instrument. Suppose we take a standard 7. We can take 4 or 9, whatever is available to us. Suppose in the beaker you have put this standard, that is pH 7. Insert the calibration rod. You calibrate the instrument to pH 7. Hence, your instrument is now ready to take the reading of the sample. In case of soil sample, we dissolve 1 gram of the soil in 10 ml of water. Stir it and allow it to settle for 10 minutes for sedimentation. Then we follow the procedure. We observe that the first sample have a pH of 7.21, while for sample 2 we have a pH of 7.24. So both these pH are ideal for sustaining of the biotic community. Do not wipe the electrodes, otherwise polarization may occur. If polarization occurs, wash the electrodes. Secondly, while inserting it into the beaker, make sure it doesn't touch the bottom of the flask. it may break the electrode.
Right.
Or it is too above the solution, then we get erroneous readings. So the best is to dip the electrode into the solution up to a mark such that the reading is stable for at least 10 seconds. Then we take the reading. It is the non-quantitative measure of the amount of organic and inorganic salts present in a sample. Require a beaker, an instrument box that has electrodes and can measure the TDS and the conductivity, a glass rod to stir, a small teaspoon to take the soil sample, and distilled water. Over here also we have to calibrate the instrument. For calibration of the instrument, we have some standards in which we dip the electrode and then calibrate the instrument using the screw. After this is done, we take the reading of the sample. We resin the sample beaker and meter twice with the water. Then we insert the stick into the container or the flask or the beaker containing the water and leave it for 1.5 minutes, and must see that the reading is stable. The TDS and EC. The total dissolved solid showed a concentration of 63.85 ppm, that is parts per million, and the electrical conductivity was 45.93 microsiemens per centimeter, while sample 2 showed 52.52 ppm and 46.03 microsiemens per centimeter. This observation tells us that the water has ions, a lot of ions dissolved in it, and it is beneficial in this case because it is the nutrients that are present in the estuarine water. This TDS and the electrical conductivity is good and ideal for the growth of microorganisms in the environment. The precautions I have already repeated before, it's the same. Salinity is the measure of the concentration of salt in water. The salinity concentrate of water causes light to refract, hence the name refractometer. It is a simple portable instrument that can be carried easily in the pocket to the field for enumerating the salinity of a sample. Normal seawater has a range of 30 parts per thousand ppt. Refractometer, distilled water, tissue paper, and the sample. This is a refractometer. For calibration of the instrument, we use distilled water. Distilled water should give a 0 reading. Over here, it should show 0 ppt. We have to wash it with distilled water until the 0 reading comes. Otherwise, there is this calibrating screw over here, which you can see. Adjust it to get this 0 reading. Now your refractometer is ready to measure the sample. A little portion of seawater is added over here. You can take off this flip, put some drops of seawater and then put it down and observe it towards the light. There is an eyepiece over there with which you can actually adjust to get a clear view. The salinity of the sample 1 28 ppt and sample 2 28 ppt. The instrument should be showing 0 ppt when tested with distilled water, and adjust the eyepiece to get a sharp image. Otherwise, there would be erroneous results. We are coming to the conclusion, and we can say that the Indian Sundarban is one of the most productive ecosystem. We have known about the physical parameters that influence the ecosystem, the functioning of the ecosystem, such as the salinity, the pH, the TDS, and the conductivity. The phytoplanktons as the producers and as a source of sink for CO2, and the bacteria as decomposers of this ecosystem.